All right. Hi, everyone. My name is Jennifer Brum, and I'm here today to talk to you about the ecosystem effects of viruses in marine oxygen minimum zones. So I am an assistant professor at Louisiana State University, and that means that I live in Baton Rouge, Louisiana. So we are getting hit with Hurricane Ida today, and I most likely have no electricity. So I'm recording this presentation and hopefully it goes okay. All right, let's get to the science. So I like to say that microbes rule the world, but viruses rule the microbes. So we all know that the ocean covers the majority of our planet, and those oceans are absolutely teeming with microorganisms. Those microorganisms are performing the biochemical reactions that really fuel biogeochemistry on this planet and help it be habitable for us. But we also know that the oceans are teeming with viruses. Viruses outnumber bacteria by approximately 10 to 1 in the world's oceans. And that means that there's approximately 200 million viruses in every mouthful of seawater. So obviously that kind of freaks people out, especially right now. But those viruses, they're not infecting you. They are primarily infecting bacteria. So there's estimated to be about 10 to the 23rd viral infections of bacteria occurring per second in the world's oceans. And all of these infections of bacteria are going to have ecosystem impacts. I'm showing you images right here to illustrate this. These are transmission electron microscopy images. On the left, we see viruses replicating within a living bacterial cell, and those viruses are having an effect on that cell's metabolism right there. But then for those viruses to get out of the cell, they're going to lyse that cell and burst it open in order to release the viruses. So these viruses have just converted this living cell into this non-living organic matter. And all of this is going to have effects on the ecosystems that we study. So the goal of my research is to understand and eventually predict how viruses affect marine systems and the Earth system in general. Now, for today, I'm going to be talking about some of the work that we've done to examine viral ecology and their roles in biogeochemical cycling in marine oxygen minimum zones. So this is a figure that my wonderful postdoc, Andrew Long, has created. This is oxygen concentration at 300 meters depth in the world's oceans. So this isn't at the surface, this is at 300 meters depth. Here we have all of our continents in gray, and here in the colors we have oxygen concentration, where you see oxygen that is 0 or close to 0 is in purple. So we have these vast regions of the world's oceans that have little to no oxygen down in the middle of the ocean. And when we take away oxygen, we have unique biogeochemical cycling because, of course, there are microbes that are adapted to exist at low or no oxygen concentrations. So this is important for the biogeochemical cycling of nitrogen, of sulfur. This can result in the production of greenhouse gases like nitrous oxide. And these oxygen minimum zones are expanding over time. They're expanding both horizontally and vertically, such that some of the oxygen minimum zones even overlap with the photic zone. So when we go out to examine viruses within these regions, what are our options to do this? Well, first of all, I forgot I was going to explain how oxygen minimum zones form. And I'm doing that because I know a lot of people in the audience are not oceanographers. So here I'm giving an example of how an oxygen minimum zone could form along the coast. Like, for example, those large oxygen minimum zones that are off the coast of Mexico. So here I have a little drawing I've made in brown. We have land. This is the beach right here, my imaginary Mexican beach house. And here we have the ocean offshore. So oxygen minimum zones really require a combination of biology and physics. In this area, we have upwelling. And so that means that this high nutrient water from down deep is coming up into the surface ocean. So when you get nutrients and light, that combination, you end up with high productivity. That's because these photosynthetic organisms that I'm showing in green dots here, they thrive on high nutrients and high light. So this area is highly productive. We get lots of fisheries. But of course, all living things eventually die. And when these organisms die, they form particles that sink to the sea floor. So these particles, as they're sinking, they're being colonized by heterotrophic bacteria that are breaking those particles down. So as those heterotrophic bacteria break down the organic matter in those particles, they are consuming oxygen. So we can get depth profiles of oxygen that look like this, with high oxygen in the upper part of the ocean because it's mixing, it's in contact with the atmosphere. And then we get low or no oxygen within the middle of the water column because the heterotrophic bacteria have broken it down. And also we're not getting high oxygenated water mixed into this area. So this combination of biology and physics, it can result in oxygen minimum zones forming wherever there are high nutrients and low mixing of that water column. So this can happen in places like upwelling regions that I just described. It can happen in river outflows, for example, the seasonal hypoxic zone off the coast of Louisiana from the Mississippi River. And it can happen in shallow coastal inlets. So we're studying several of these different types of oxygen minimum zones. We're examining viruses and their viral infections of bacteria in 7 different regions that I'm showing here. So we have the Arabian Sea, we have the large oxygen minimum zones of the Eastern Tropical North Pacific and Eastern Tropical South Pacific. We have the hypoxic zone in the Northeast subarctic, and we have Saanich Inlet, a little inlet here, the Gulf of Mexico hypoxic zone, and the Cariaco Basin. So we go out and we start to examine viruses in these areas, and some of the tools that we can use are visual. So I can count the total number of viruses and bacteria in my samples using epifluorescence microscopy. So this is what seawater looks like over here when it's been filtered onto a 0.02 micron pore size filter and stained with a nucleic acid stain. If we look at it under an epifluorescence microscope, we see these larger dots, which are bacteria, and we also see all these numerous smaller dots, which are viruses. So now I can count viruses in my samples. I can also use electron microscopy to look at the morphology of viruses in the samples. I can see that viruses come in various different shapes and sizes, some with tails, some without tails. And I can also use electron microscopy to examine the infection frequency of bacteria in these samples. So here I'm showing different types of bacteria right there. You see those viruses, those bacteria have no visible viruses within them. Whereas these bacteria right here clearly contain visibly replicating viruses. So we can do all of these things to examine viruses in the ocean. But we have a problem. When I'm using this, I can't tell what types of viruses that they are. What genes do they have? What hosts do they infect? What is their replication strategy? All of this matters when I'm trying to figure out what effects on the world this little glowing dot is going to have. So we also have an issue that viruses have no universally conserved gene. So there's nothing equivalent to a 16S gene or an 18S gene like you would use for other living organisms. So how do we even compare samples when I can't identify what's in one sample? Well, our solution to that has been to investigate viral diversity using metagenomics. So I'm showing you the general path of metagenomics here on the left. Our first step is to obtain some seawater, and then we're going to concentrate viruses from that seawater. We use iron chloride flocculation to get the viruses to stick together based on that iron chloride addition. So now we have our concentrated extracellular viruses. We then extract the DNA from those viruses and we amplify the DNA. We use linker amplification, which is the most quantitative way we know how to amplify DNA. After that, we'll sequence the DNA, for example, using Illumina sequencing for large metagenomes. And then we analyze those sequences to obtain amazing knowledge of marine viruses. And my favorite way to do that is to work with Simon Rue at the Joint Genome Institute. So hi, Simon. So one of the most important things that's happened to help us be able to analyze these sequences is that we now have definitions for viral populations. So that means that if I look at the genome sequence space of all the different viruses in a sample, I see that they can sort of cluster together. So we can examine our viral metagenomes by looking at contigs that are assembled greater than 10 kb, by clustering those into groups which are over 95% average nucleotide identity, and then annotating the genes. After this, we run it through Vir sorter to determine, well, is our best guess, is this a virus or is this not a viral genome? So now we can quantify viral populations in different samples. I can say that this virus population is more abundant than this population, and then I can compare them among different environments. So that's what I'm showing here. This is work done by Andrew Long for the global analysis. We have sufficient viral metagenomes from 5 different regions. So I've got each of those 5 different regions here on the y-axis, and I have those same 5 different regions here on the x-axis. What we're looking at is the presence of various different viral populations we've detected, where they were first detected versus where else they're detected around the world. So the first thing we notice here is we have 3 regions right here, which all have more overlap than the other ones. These are the Eastern Tropical North Pacific, which is permanently anoxic, the Eastern Tropical South Pacific, which is permanently anoxic, and the Arabian Sea, which is also permanently anoxic. So these regions, you cannot measure oxygen with them, and they do not mix during the year. So we're seeing that the Eastern Tropical North Pacific and South Pacific are a little bit more similar, whereas the Arabian Sea is a little bit more divergent from them, but it's half a world away. Then we go to the Northeast Subarctic Pacific, and we see that that is divergent from the other oxygen minimum zones. This is a permanently hypoxic region. So that means it doesn't mix, but the oxygen concentration only gets down to low levels of oxygen. Finally, we have Saanich Inlet up here, clearly having very divergent viral populations, high endemism, and that is a coastal seasonally anoxic inlet. It mixes every single year, but when it doesn't mix, it becomes sulfidic down in the anoxic region. So that means you get a buildup of sulfide as well. So I want you to compare this right here, where we're seeing these divergence of viral populations found in each of these regions, a lot based on how that oxygen concentration is in each of these regions. So anoxic, hypoxic, or seasonally anoxic. But now compare this to the viral gene functions in each of these regions. So I'm showing here, there are 5 different regions: Saanich Inlet, Subarctic Pacific, etc. And we're looking at the abundance of the protein families in each of these gene categories. And we're seeing similar gene functions in every single region. There's no clustering like in the previous heat map. For some of this, it's pretty easy to explain. Things like replication and lysis, or structural genes, or transcription and translation. Those are things that every single virus needs to replicate. However, we also see this category right here, which is metabolism. So we're seeing metabolism genes in each of these regions. And some of you might be wondering, why would a virus have a metabolism gene? It's not alive. So for that, we go to the fact that viruses can have auxiliary metabolic genes. The definition of this is any metabolic gene that a virus has that's not directly involved in viral replication. So the classic example here is a photosynthesis gene. Here we have an illustration that I've made of a prochlorococcus cell. It's a photosynthetic organism. It's up in the upper ocean consuming carbon dioxide, and it's producing oxygen through photosynthesis. This requires a gene called PSBA. Now the virus that infects this prochlorococcus also has the gene PSBA. So when that virus infects that cell, it injects its DNA and it expresses that PSBA gene. This increases photosynthesis within the cell. But the virus is not doing this to be nice to the prochlorococcus. It's doing this because it needs to make more viruses, and it requires energy and materials to make all of those viruses. But in this process, viruses are directly affecting photosynthesis in the ocean. They've increased the amount of carbon dioxide consumed and increased the amount of oxygen produced. So it's not just photosynthesis genes. Viruses have also been found to have nitrogen metabolism genes, sulfur metabolism genes, genes involved in phosphorus acquisition, carbon metabolism. Keep going, and we've probably found it in a virus. So our question here is, what are these metabolism-related genes that we're finding within these regions that have oxygen minimum zones? For that, I'm going to give you an explanation from the Eastern Tropical North Pacific, one of those large permanently anoxic regions. And it has the best depth profiles that we have for any of our study regions, so we've studied it more intensively. So here I have a depth profile from the Eastern Tropical North Pacific. We're going from 0 down to 1,000 meters depth down in the ocean. In the green, we have chlorophyll fluorescence. So we have our photosynthetic organisms up at the upper part of the ocean. And in blue, I have oxygen concentration with the highest concentrations of oxygen at the surface, and then decreasing down to almost nothing below 100 meters, staying low all the way through the water column down to about 800 meters, where it starts to increase again. So first we use our tools, our visual tools. And for that, this is work that I'm showing you from Sarah Schwenk, who is now a graduate student at Scripps, and also Sophie Jurgensen, who is a graduate student in my lab. So if we use the tools first to count viruses using epifluorescence microscopy, and we see that viruses are in fact present. Here's our depth profile of virus concentrations, higher in the upper ocean, decreasing down with depth where there's little oxygen. It looks like there's almost no viruses there, except for the fact that this x-axis is on a scale of 10 to the 7th viruses per mil. So we actually still have a lot of viruses within the oxygen minimum zone. Then we can look at the frequency of infected cells using electron microscopy, and we see that viruses are in fact killing bacteria within the oxygen minimum zone. The frequency of infected cells is high in the upper ocean, decreases here around the oxycline, and then increases again within the oxygen minimum zone. So the amount of infected cells within the oxygen minimum zone is actually similar to up in the upper ocean. Finally, we get to morphology. We can look at the types of viruses that are found there, and we see that most of these viruses are non-tailed. So here I'm showing you samples from various different depths that we collected. And the most viruses that we see is these light blue bars, which are these non-tailed viruses that have no tails. So they actually increase down when we get to the anoxic region. It's primarily dominated by non-tailed viruses. So this is great, but now we want to look at our metagenomic results as well. So right here, I'm again showing you the exact same depth profile, and I'm showing you an ordination plot comparing the viral communities in each of the regions from our, in each of the samples from our study. So here we have oxic samples. Those are the viral communities in the oxic regions from 2 different stations, clearly different amongst those stations. But then we compare those to the viral communities where there is low or no oxygen, so throughout the rest of the water column. And we see that they are highly similar to each other compared to the upper ocean viruses. Now we're going to zoom in a little bit and look at how those viral communities compare to each other when you just examine the low oxygen communities. So that's what I'm showing here. Right here, we have the viral communities from the oxycline. That is where that oxygen concentration is going down to 0. Here we have viral communities from the region without oxygen at 300 meters. And here we have viral communities from below the oxygen minimum zone where oxygen starts to increase. So clearly we're seeing that viral communities are affected by oxygen concentration. But we also note that these viruses are still diverse as we go down through the water column. So the differences between viral communities with and without oxygen is not that we have lower diversity without oxygen, it's just that we have different viruses there. So now we start to get to the meat of this. We want to look at what viral auxiliary metabolic genes are in our samples. They're in our Eastern Tropical North Pacific. So I go to Sophie and I ask Sophie, please show me a depth profile of viral photosynthesis genes at this station. And she shows me this. Viral encoded photosynthesis genes are highest in the upper ocean where there is sunlight and where there is photosynthesis going on. So this is good. This means the world is working the way we think it should. Then I say, Sophie, please show me these beautiful depth profiles of viral encoded nitrogen and sulfur metabolism genes, because those are some of the main biogeochemical cycles we're really interested in. And she shows me this. We have very, very low abundances of these nitrogen and sulfur related genes. And I'm showing these on the exact same scale as for the photosynthesis genes. So why are the viruses in oxygen minimum zones not containing these vital, what I think should be vital, metabolism genes related to nitrogen and sulfur? We think that's because a viral genome must fit into the capsid. So the genome of this virus has to fit in this capsid. It's like fitting things into a box when you're moving. That means that there's going to be intense selection for only including genes that increase viral fitness. If that gene does not increase the fitness of that virus, it is going to be selected against. So our hypothesis from this is that the host cell metabolism is already sufficient, that the host cell's metabolism of nitrogen and sulfur is already enough for those viruses to replicate, and those viruses having those genes related to nitrogen and sulfur is just not going to benefit them. So this is very important research. This is very important finding, but we are very disappointed. So we still have this same question. What are these metabolism-related genes that we've annotated within our oxygen minimum zones? So for this, we remember that viruses have other ways of manipulating their hosts other than the central metabolic cycles. Sophie comes to me with 3 graphs of 3 different gene types, different gene groups. We've got sporulation genes here, iron sulfur cluster genes, toxin antitoxin genes, and all of them have suspiciously similar depth profiles. They're low in the upper ocean. They have their peak concentration around the base of the oxycline, right here where oxygen is going to 0. Second highest abundance within the oxygen minimum zone core, and the lowest abundance down at 1,000 meters. So I told Sophie, go start getting reading. Why would viruses require these genes within an oxygen minimum zone? So she starts reading things and she finds that these sporulation genes have been found in viruses to induce the sporulation in their facultative or obligate anaerobic hosts. Then she finds that while iron sulfur cluster genes can be used in a lot of different things, they have been found to be involved in dealing with oxygen stress. And finally, she finds papers showing that toxin antitoxin genes in viruses have been used to keep a stressed microbial host from kicking out a viral plasmid. So all of these things seem to be involved in dealing with oxygen stress. And this makes sense because their lowest abundance is in the upper ocean where there's plenty of oxygen, or down deep where there's oxygen. Their highest abundance is right here at the base of the oxycline, where oxygen is going from higher usable levels to low levels. And this could stress out the host cells. So our conclusions based on this data is that yes, in the upper ocean, we are seeing photosynthetic genes within viruses. When there is sunlight, when there's oxygen, the viruses will have these photosynthesis genes to increase production of viruses from their hosts. However, the viruses in these OMZs apparently don't need high concentrations of directly related to nitrogen and sulfur cycling. Instead, they seem to have genes that are used to keep their host alive and healthy up until they can kill it. So Sophie has illustrated that here by showing these shields here, this virus expressing genes that will protect its host from stress up until the point that it can replicate enough viruses and then kill the host. So this could increase the fitness of that virus within oxygen minimum zone regions. So I'd like to conclude here by talking about our next research and looking really at the overview of how viral replication works. So this is the basic overview. Our virus meets its microbial host. The virus replicates within the cell. And then if it's using the lytic pathway, it's going to lyse that cell, burst it open, and produce extracellular viruses. So this is what we have been examining. How much lysis is there? How many bacteria are the viruses killing? And we've been examining the extracellular viruses. How many different types of viruses are there in our different samples? But what we need to do is what we're currently doing. We need to investigate this part over here where the virus is interacting with its host bacterium. We need to know what viruses are inside the cells within these regions and what genes are they expressing while they're in those cells. And finally, we need to examine, are they using lysogeny? Are they just residing within their host cells until something causes them to switch to the lytic cycle and kill their hosts? So I will thank you for your attention today, and I would like to acknowledge first our funding sources, the National Science Foundation and the Department of Energy Joint Genome Institute for doing all the sequencing for this. I'd also like to acknowledge my collaborators, Simon from JGI, Frank Stewart, Stephen Hollum, Osvaldo Ulloa, and Matthew Sullivan. And finally, I'd like to acknowledge my lab. We call ourselves the Go Viral Lab. That is Cajun French, if you're wondering. And of course, you've met my postdoc, Andrew, my PhD candidate, Sophie. I have another PhD candidate, Ariel, who is working on very challenging samples from hydrothermal vents, and my lab manager, Emily Savoy. And here are my undergraduate researchers as well. So everyone, I hope this recording was understandable. Please reach out to me via email if you have any questions or comments. Email is going to be a little bit slow though after this hurricane. So I hope you are well, and thank you very much for listening.